So we were talking about the general base. Sorry, so we were talking about. So I was talking about the fact that the major thing that an enzyme does is to what is to try to bring down the activation energy by making the transition state more stable altogether. But we are saying that in a general base catalysis now, in a general way catalysis, what happens is that a base will be used to abstract, or let's say to remove one proton from this intermediate in order to make this particular thing more stable. So that's what happens today. So when you can now say that a general base catalysis involve a general base catalysis is a mechanism of catalysis that involves using a base to abstract a proton, to abstract or remove a proton from the transition state, thereby lowering the energy of the transition state, at the same time the activation energy. Now, so most times when if you are going to look at, or let me put it that way, that most times if you are going to have either a general acid or a general base, it most times involve probably if the transition state is some that can probably release proton, or some that can probably collect protons. So the reason why we can have now, for example, now 9%. So for example, now we might not be able to stabilize this O altogether now, because in a way it doesn't really have any proton. We might have to use another method to stabilize this Mr. Man altogether. So it is not, but most time if you are going to use your general acid or your general base, it must be that probably a part of the intermediates can either release proton or accept protons. So in this case, you can use an acetate ion. You can use an acetate, which is C double bond O. Sorry, C single bond O minus, and we have double bond O, and we have R. This is an acetate ion, and this in a way we do can collect this can withdraw this proton, and this thing becomes stabilized. Do you get that? So because a base is involved, that's why I call it a general base catalysis. Do we get that? You can ask a question if you have.
No question.
If you have questions, you can ask them, because don't worry, I will try to go any paragraph. So that is for a general base catalysis. So let's talk about the one for the general acid catalysis, because in some cases you might have to use an acid that probably donates a proton in order to keep the intermediate more stable, at the same time lowering the activation energy. So now let's look at that example. Can I rub this?
Yes, sir.
Okay. Don't let me. I don't want to. So just permit me to use this. So let's look at this now. Let's look at this structure. We have O, C, we have OR. So this is hydrolysis of an acetal bond. This is hydrolysis of an acetal bond, ACET. This one is as, this one I said is an ester. This one is acel. Yes, but you can also hydrolyze this. So, but in the process of hydrolyzing this, you can, the point is that when you hydrolyze this now, this one will form what? ROH. But this one will form this, the OH will come to what? H will be added to this. OH will come, H will come to what? Will come to this. It's like probably when you have two sugars bounded together and you want to hydrolyze it. The bond between two sugars is always this old glycosidic bond. You get that? So if I want to hydrolyze them now, H will go to one part, OH will go to another part. You get that? So, but this is just like a similar thing that we have here. So if I want to hydrolyze this now, an intermediate also will be formed in this case. So the intermediate form from this is always of this nature. We are going to have, so we are going to have this over here. So this will be partially positive, and you have dash and what? OR. And this one will be partially what? Negative. So the formation of these charges makes this intermediate what? Unstable. So one way by which, so in a way to make the reaction more faster by an enzyme, what an enzyme will do is to what? Is to bring the what's it called now? The energy of the transition state to what? To lower the energy of the transition state. So, but in this case now, I'll get that. In this case, we don't have anything that can work. We don't have anything that can work that can lose a proton. So one way by which the enzyme work is to what? Is it can probably what? Stabilize this negative charge. So in the first one that we did, the enzyme stabilized what? A positive charge. And how does it do that? It remove a proton. Do we get that? But in this case, what the enzyme will do is to stabilize Now, the enzyme may not be able to stabilize this one because at the same time there is no this one does not ionize. It cannot lose a proton as we have for water in this case. Water can easily lose a proton and then we have the OH. Are we clear? But in the case of this positively charged carbon here, this one does not have an ionizable hydrogen. Do we get the difference now? So what the enzyme will do here is to stabilize the negative charge by by adding a proton to it. So and that means that in that case you might need you might need an acid that can donate a proton. So in the case of this, so in this case now, you can have you can have an acetic acid, that's H. For people, please bear with me so that probably the board we have. Let me write it somewhere, please. Let me put H. We have O. Sorry, we have O. We have C double bond O, and we have R. So in this case now, so this hydrogen that you have on the acetic acid, the hydrogen of the acetic acid can be used to stabilize this negative charge. So and this thing is full of electron. It has been much electron. That's why it's negative charge. So if you want to stabilize it, you have to add a positively charged system to it. So but that positively charged is our proton that's coming from an acid. Altogether. So when you are stabilizing a negatively charged entity like this in the transition state, we call it using an acid now, we call it a general catalysis. Altogether. So when the process of stabilizing, or let me say lowering the energy of the transition state involve donation of proton by an acid, when the stabilizing process of the transition state involve donation of proton by an acid, or let's say it involves proton transfer from an acid. When it involves proton transfer from an acid, it is called a general acid catalysis. Yes.
So for the first flow,
yes.
the example you drew from water to I think the ester. What does that denote?
This line, right? it implies that it's just to show the thing is bonds in the transition state, they are neither formed nor broken. Do you get the point now? The bonds in the transition state are neither formed or broken. So one way by which we indicate a bonding in the transition state is by using what? By using this dash. It's just to show us that this thing that we have, this system that we have here is what? Is a transient entity that doesn't spend a word, that doesn't have much time to spend. So this bond between this O and this C is a transient bond, and that's why we are using what? We are using dash to indicate it. The same thing applies to the one I show. Yes.
So that means for the first one, which is a base catalysis, the base is donating
a base is removing proton. Yes.
So is water the base in this case?
No, the base in this case is acetate ion. I mentioned that earlier. That you are having now water will not be the base in this case, because the thing is if I want to talk based on from our knowledge of acid and base, I will say that water in this case is the acid. This part of the molecule is the acid because it is the one losing the proton. But the collector of that proton is an acetyl ion. That is your R. This is O minus, and we have C double bond O, and we have R. So it is this acetate ion that will collect. Yes, it is this acetate ion that will collect the proton from this Mr. Man. Do you get that?
So the acetate ion is the base.
Yes, it's the base in this case. But the acetic acid is this one that we have here is what? Is the acid here. So when you are trying to stabilize the intermediate using an acid, when you stabilize the intermediate, or let me say when you not the intermediate now, when you stabilize the transition state with an acid that is donating a proton, that's what? A general acid catalysis. But when you stabilize the transition state with a base that is collecting that is accepting a proton, that's a general base catalysis. Do you get that?
Yes, sir.
Okay. So the next one now, the acid. Normally we always have acid base catalysis. So we can now say that acid base catalysis can be general and it can be it can be specific. I've talked about the general acid base catalysis, both for the acid and then, but we also have the specific acid base catalysis. Now, what difference does it make between the general ones and the specific one? The difference is that in the specific acid base catalysis, H plus hydrogen ion and hydroxide ion, or let me say water is the water is the acid donor. Sorry, water is the base. At the same time, water is the acid. I don't know if I'm making sense. Now what I'm trying to say there is that if you are using water, you are using water in place of in place of this acetyl ion, or you are using water in place of this in place of this acetic acid, that becomes a specific acid base catalysis. Do we get that? So in some cases now, your proton acceptor here might be water itself, and if water accept proton, what does it become? It becomes H3O plus. Now at the same time, it can also be It can also be the acceptor here. So, or let me say the donor here can also be water. So I was saying that if what we have is, so if you are having water in place of an acid, or you are having water in place of a base. Now in some cases also you can say that this one is OH minus out there, and this one is H plus, but it still implies that you are using water. You are using water to either be a proton donor or the proton acceptor. So that is when you have a specific acid base catalysis.
Okay, so sir, that means that acetate
Yes, yes. At the same time,
So you have water from transition state, and then you have water act as the base again.
yes. That means the thing is the reaction itself involve the hydrolysis of the ester bond involve water itself in forming the transition state. But we are only saying that in a specific acid or base catalysis, water will still be the one that will do what, that will be the what, the proton collector or the proton donor. Do you get that? If it is probably you are trying to stabilize a positive charge, it will be the what? It will be the proton acceptor. But if you are trying to stabilize a what? A negative charge, it will be the proton donor. But if you are using another thing apart from water, that's where we what? A general base or acid what? Catalysis. Now the next one that we also want to talk about is that we want to talk about the electrostatic catalysis. Electrostatic catalysis. Electrostatic catalysis. Now this in a way occurs. This occurs when the charge part of the transition state, when the charge part of the transition state does not have when the charge parts of the transition state does not have an ionizable proton, or let's say ionizable hydrogen. And I've talked about something like that the other time when I was trying to make a difference between this positively charged here and positively charged that we have here. That the positively charged system here has a what? An ionizable hydrogen. But the carbon that we have here does not have an ionizable, so it cannot be stabilized by what? By a what? A base. Altogether, by a base what? Withdrawing a proton from the what? From this place. Do you get that? But on the contrary, the way by which an enzyme will stabilize this positive charge is by using what? Is by the electrostatic, what's it called now? Catalysis. And that involves stabilize. Now you can write it down. It involves stabilizing a positively charged system. It involves stabilizing a positively charged system with a negatively charged system. Now in this case you are not removing protons to stabilize it. How do you do that? You are not removing proton, but you are trying to use your electrostatic interaction, which involve, there are times that the positively charged entity might not, or let me say this transition state might not have an ionizable hydrogen to probably be donated to what's called to a base, or probably cannot even accept what's called now a proton from an acid. So in that way, enzyme still uses another way to try to bring down the energy of the transition state. And we are saying that they can also use electrostatic interaction between a positively charged part of the transition state and another a negatively charged ion altogether. So in a way, what I'm trying to say there is that this positively charged carbon can be stabilized with either your acetate ion. But in this case now, it will not be as though you are donating It will not be as though you are donating a proton to this acetate ion, but rather you are having an electrostatic interaction between this O minus and C plus. Do we get that? So when you have such interaction that brings down the energy of the transition state, then you can say you have an electrostatic catalysis. Do we understand that? So and then you can write it there that in the hydrolysis, so you can now put it that, for example, in the hydrolysis of this acetal bond, the positively charged carbocation can be stabilized. The positively charged carbocation can be stabilized with a negatively charged carboxylate ion. can be stabilized with a positively charged carboxylate ion. Can be stabilized with a negatively charged carboxylate ion. Can be stabilized with a negatively charged.
Yeah, which one is that for? Is that for electrostatic?
For electrostatic, yes, catalysis. For electrostatic catalysis.
Sorry, I didn't get it to.
From which point?
The hydrolysis of acetyl.
I said.
What you just dictated.
Okay, I said that parts, when positively charged part of the transition state is stabilized by a negatively charged ion. That's when you have like, and that interaction is involving an electrostatic interaction. That's when we have an electrostatic catalysis. I now said that an example of that can be seen in the hydrolysis of what? Of this acetyl bond that we have talked about earlier, whereby the positively charged carbonium ion or carbocation now, the positively charged carbocation is stabilized with a carboxylate ion. So in that case now, the positively charged entity is not donating what? It's not releasing what? Any proton altogether. But what is keeping the transition state stable is what? Is the electrostatic interaction between the what? Between the negatively charged carboxylate ion and the positively charged what? Carbocation. That's what we have here. Now you can also have it in the same manner that a positively charged ion is stabilizing a negatively charged, the negative charged part of the what? Of the transition state. Altogether, as you have that a positively charged ion can stabilize the positive parts of the transition state. You can also have that a positively charged ion, you can also have that the positively charged ion is stabilizing the negatively charged parts of the transition state. And that process is what we refer to.
Yes,
Yes. So I said that process is what we refer to as an electrophilic catalysis. The process of stabilizing. Yes, I said the process of stabilizing a negatively charged part, of the transition state. The process of stabilizing the negatively charged part of the transition state. The process of stabilizing the negatively charged part of the transition state using a positively charged ion. You can put in bracket most times meta ions. Most times meta ion. Is what we call the electrophilic catalysis. You know what electrophiles are? They are what? They are electron what? Lovely. Yes.
So, that means, is it that nucleophilic stuff is not when a positive part is stabilized with negative charge?
Yes. I will still talk about the nucleophilic part too. I will talk about it. But the first one I talked about, it is not nucleophilic, because what we talked about initially, you having a negatively charge balancing a positively charge on the transition state is more electrostatic than you having what? A nucleophilic interaction. Most times when you have something, when an enzyme try to lower the energy of a transition state by a nucleophile, it always involves probably a formation of what? Of a bond. altogether. It's not like something that you now what we're trying to say here, in the first case that I mentioned, I talked about what electrostatic what do we call it now? Electrostatic constant that you have here is like what you have between sodium plus and what and Cl minus. Altogether, that interaction is not what? It's not a nucleophile electrophile thing. It is just your normal ionic interaction. That's what you have. The same thing is what we are talking about in this case. In the first case, whereby you are having carboxylate ion balancing the what? Or let me say stabilizing the what? The positively charged cation here. So the first one I talked about is more of electrostatic bonding than you having a what? A nucleophile. Yes, we know that it is we know that what is balancing that this positively charge is a nucleophile. That one is because it is what it is nucleus loving, but at the same time the interaction there is more of an electrostatic interaction than you having a nucleophilic thing. Okay, we are still going to talk about a nucleophilic catalysis too, but I just want us to, I'm just following what we have in this particular place. Do we understand that, please? Okay.
This is more of what is coming into my brain.
Okay, yeah.
Okay, what I was thinking was all this donation and accepting,
Okay.
like accepting and there's like the enzyme has like composed of protein.
Yes.
And this protein has amino acid,
Yes.
the acidic and some
Wow.
bases. So in this kind of scenario, like can't those, instead of just getting other molecules, maybe water or like in this case, you see,
Yes. Yeah.
instead of that one, can't you just like
Use their own. Yes. The thing is, it is not every. Now, when we say carboxylate ion here, or when we say acid here, now in the protein itself, all of these chemical entities that we're talking about are not usually from the outside.
Mm-hmm.
Do you get that? Because and that's what probably brings about what we call the active sites.